Okay, so let's continue on slide 7, skeletal muscle, which is where we're going to spend most of our time. And this is voluntary striated muscle. So we control skeletal muscle, and it's striated. Looks striated under the microscope, and so does cardiac muscle if it's in the longitudinal section. But cardiac muscle is not voluntary, so cardiac muscle will be involuntary striated muscle. The cells are long, cylindrical, multinucleate cells called muscle fibers or myofibers. Sometimes called myocytes. They're microscopically thin, but macroscopically long. They can be like a foot long or more, depending on how long the muscle is. Um, but the diameter of the tube is microscopic. They're from the fusion of myoblasts, which are fetal muscle tissues. And some fetal muscle tissues still exist in adults as what are called satellite cells. And they can replace myofibers to a limited extent, but generally they don't. Generally, you don't get skeletal muscle cells back if you lose them. And if you go to the gym and build up muscles and make really big muscles, you're not actually adding cells to it. You are just adding to the size of the cells that are already there. So this next slide 8 just shows that these fetal tissues called myoblasts fuse to form a skeletal muscle fiber, which is a long cylinder, and it has multinucleate. It's got lots of nuclei. It's one of the few multinucleate cells in your body, besides osteoclasts, which are also multinucleate. And the reason they're multinucleate is because they have to make proteins up and along this long, macroscopically long cylinder, and there's no way they can move things efficiently or fast enough if there wasn't a lot of nuclei making the proteins. So the muscle fiber, slide 9, um, the muscle cell, and the prefix for muscle is sarco. So sarcolemma is the cell membrane. Sarcoplasm is the cytoplasm inside the muscle cell. And there's glycogen stored in muscle cells, and there's something called myoglobin, which is similar to hemoglobin, but is a little tiny oxygen tank essentially inside muscle cells. There's the endoplasmic reticulum in skeletal muscles called the sarcoplasmic reticulum. It has these things called terminal cisternae, transverse tubules, and triads. We're going to get to those in a moment when we look at a picture of the inside of muscle cells. And the sarcoplasmic reticulum is a storage area for calcium, which is the trigger for muscular contractions. Then myofilaments are parts of the muscle cell that are composed of proteins, and we're going to have to learn several different kinds of proteins, and that make up what are called thin filaments, thick filaments, and elastic filaments. So we'll learn muscle proteins called myosin, actin, tropomyosin, troponin, titin, and connectin, and a couple others along the way. Here's slide 10, which is a drawing of the microscopic anatomy of a muscle cell. And we have these myofilaments inside the myofibrils. So myofibril is a bundle of myofilaments, and a bundle of myofilaments makes up a muscle cell. This is part of the bundle of bundles. And then there are the sarcolemma here, which carries the action potential, and the action potential is carried down deep within the cell by what are called the T-tubules. And then the terminal cisternae of the sarcoplasmic reticulum are along the T-tubules. And we're going to find out that the action potential, when it's brought down inside the muscle cell, it's going to open up calcium ion channels and allow calcium to leave the sarcoplasmic reticulum. And down here in part B, we'll see we have sodium-potassium pumps like we have in all cells, voltage-gated sodium channels, voltage-gated potassium channels, just like in nerve tissue. And the action potential is brought down into the tubules, deep within the muscle cell. And this opens up voltage-gated calcium ion channels and allows calcium to leave the terminal cisternae and enter the sarcoplasm. Okay, slide 12, the proteins in the SR and SR membrane, the sarcoplasmic reticulum. There are, as I mentioned, voltage-gated calcium channels in the sarcoplasmic reticulum, and there's calcium pumps. For one muscle relaxes, you have to turn on calcium pumps to get to put the calcium, resequester it back into the SR. And there are proteins within the SR called calmodulin and calsequestrin, which actually bind calcium. Then we have two types of proteins in the myofilaments, what are called contractile proteins, myosin and actin, which actually allow the muscle, which actually cause the contraction in the muscle. And then we have regulatory proteins called tropomyosin and troponin that control the contraction. And skeletal muscle is striated because it has sarcomeres. And these are the sarcomere is the smallest part of a muscle that actually contracts. And it has various bands called I bands, H bands, and A bands, Z discs, amylines. We're going to look at that in the next video, second slide in the next video. And then we're going to look at in the next slide in the next video how these contractile proteins and regulatory proteins are arranged in thin and thick filaments. But I gotta stop now because I'm afraid I don't want to make sure this video isn't too long, because if it goes too long, it won't upload to YouTube. So I'm going to cut this one off right here.